Similarly, with X-chromosome inactivation, I was never able to study that full time because of my main remit was to study radiation damage, but I continued with it in my spare time with the aid of visiting workers and graduate students. My main work involved many papers on various doses and dose rates of radiation and various endpoints for measuring radiation and comparison of different species. And these papers all went to international committees which were estimating the genetic hazards of radiation. In 1962, I found myself the head of the group at Harwell, and I followed the same general plan as my predecessors had, but I also tried to broaden the types of genetics being studied in the mouse while still maintaining the main remit of radiation mutagenesis. The first new subject, new feature we added was cytogenetics. We studied X autosome translocations and male sterile translocations, and we used translocations as an endpoint in order to study the effects of radiation. Other features we introduced were biochemical genetics and mouse embryology. And the work on embryology was much assisted by me spending a sabbatical year at Cambridge in 1970 to 71. I worked with Richard Gardner, who was just then beginning his work on surgery of the mouse blastocyst, aimed at understanding the differentiation of the various germ layers of the embryo. Richard and I studied the onset of X chromosome inactivation. Richard injected one cell from one embryo into a blastocyst of another embryo, and we were able to tell from coat color markers whether X inactivation had or had not occurred at the time the donor cell was injected. The answer was that it had not, which gave us some idea of the time of onset of X chromosome inactivation in the embryo. Also working at that time was David Whittingham, who was engaged in developing a method for preserving mouse embryos in liquid nitrogen, frozen in liquid nitrogen, that is. And when he obtained success, we collaborated with him in testing the feasibility of preserving stocks of mice by freezing embryos in liquid nitrogen. And so mouse frozen embryo banking became started through that work. In addition to research, I was also involved in matters of mouse gene nomenclature. First, I was editor of the Mouse newsletter, which was a forerunner of the online databases that one has today for looking for information. The Mouse newsletter used to give lists of known mouse gene loci and of up-to-date genetic maps, and also mouse genetics research news. These lists of gene loci were obviously very useful in our radiation work. And the Mouse newsletter has now been taken over by an online database. At a somewhat later time, I was chairman of the International Committee on Mouse Genetics Nomenclature, and we arranged the publication of 3 editions of a work on genetic variants and strains of the laboratory mouse, which again gave lists of mouse gene loci and genetic maps and mapping data, and also gave lists of available mouse genes and congenic strains and DNA variants. And again, like Mouse newsletter, it's now been taken over by an online database. In 1986, I retired as head of the genetics division, and my place was taken by Bruce Catanack, who is a very distinguished mouse geneticist. And he in turn was followed by Steve Brown, and the name of the unit, the title of the unit, was changed to the Mammalian Genetics Unit, and it no longer has to study radiation genetics. I wish the MRC had made this change earlier. Since 1986, I have been engaged in various collaborations with others aimed at identifying the genes underlying the various mutations that we discovered previously. Thus, I've had the privilege of seeing and taking part in the development of mouse genetics from the stage of hardly anything being known when I started out as a student in the 1940s to the present stage of a complete known DNA sequence and thousands of identified genes. Thank you.
Thank you very much. We're going to present you with the prize in a moment. Now I ask Joan Didion to join us. Thank you, Dr. Lyon. I'd now like to invite Joan Didion to join us at the podium for the reading of the citation and the presentation of the prize. I'm going to read the citation. On this, the second day of November, the Rockefeller University honors you, Mary Frances Lyon, as the recipient of the 2006 Pearl Meister Greengard Prize, an international award recognizing outstanding women in biomedical research. In the early 1950s, when women who sought careers in the sciences encountered daunting challenges, you undertook investigations that profoundly influenced the course of biology and medicine. As a young scientist, you developed the hypothesis of X chromosome inactivation, which to this day is considered one of the definitive findings in modern genetics. Your insights revolutionized the understanding of gene regulation and hereditary diseases and gave the world a new scientific term, lionization. With exceptional foresight and determination, you pioneered the field of mouse genetics at a time when little was known about any mammalian genome. You were prescient in your resolve that by focusing on the mouse, scientists could unlock doors to understanding human biology and disease. Through meticulous studies, you helped to assess the risks associated with radiation, chemical mutagens, and other environmental agents. Taking this work beyond the laboratory, you engaged in the effort to ensure that science informs policy decisions and serves the public good. Today, in presenting you with the Pearl Meister Greengard Prize, we recognize and thank you for your distinguished leadership in the international scientific community and your many enduring contributions to biomedical understanding. Thank you. I'm now going to ask Joan if she will read a brief statement.
It is my honor this evening to present to Dr. Mary Lyon the Pearl Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the distinguished jury of the Pearl Meister Greengard Prize and all of us here today, we congratulate you on this award.
I should like to say how extremely honored I am to be given this award. And I'm particularly honored to obtain an award for women because, as has already been said, I think women don't receive a fair share of the awards and distinctions that are available in science. And I very much appreciate the generosity of Dr. Greengard in giving his money and Rockefeller University of backing him. And I should like to thank everybody concerned for awarding me this honor.
Great, thank you. You can talk, I'll just... Well, thank you all very much for coming this evening. I think this has been a wonderful occasion and a wonderful award. And I invite all of you to join us in the Abbey Lounge out there to the left for the reception. Thank you very much for coming and for being here. Thank you.